Hi, we've already learned a bit about the cell cycle and its phases, G1, S, G2, and N. Now we're going to add another layer of detail as we start to discuss how the cell cycle is regulated. In this video, we'll talk about cell cycle checkpoints. First of all, why is it critical that the cell cycle be regulated? Well, if a cell is replicating with mistakes, either mutations or the incorrect number of chromosomes, we could have major problems. One of the most significant consequences of dysregulation of the cell cycle is cancer. So internal controls on the cell cycle are really important to prevent cells from dividing uncontrollably, which is a major hallmark of cancer cells. Here's the cell cycle again. We're already familiar with the progression of the phases, from G1 to S to G2 to M phase. Now we're going to add 3 checkpoints. Checkpoints are a time in the cell cycle in which the cell cycle can be arrested or stopped. Sometimes a cell will stop at these checkpoints temporarily and then be able to move on. Other times it will not, and we'll talk about what happens in that case just a bit later. So let's talk briefly about the checkpoints. The first checkpoint we will discuss is in the G1 phase. Recall that in G1, the cell needs to irrevocably commit to proceeding through the whole cell cycle or to leave the cell cycle and go into G0 phase. If the cell is committing to continuing through the cell cycle, it needs to be large enough to divide again and have enough resources. And perhaps even more importantly, it needs to check for DNA damage. We most definitely do not want cells with significantly damaged DNA to be moving through the cell cycle, because DNA damage can cause mutations, and mutations can eventually cause cancer. If DNA damage is detected, the cell will attempt to repair it before moving on to the next phase. Okay, so let's imagine that our cell has passed the G1 checkpoint and moved on to S phase. Hopefully you remember that DNA replication takes place in S phase. And then we move on to G2. There's another checkpoint in G2 that we're going to very briefly discuss. In G2, the cell really wants to make sure that the genome has been completely duplicated, and also that our DNA has not been damaged in the process of replication. If a problem is detected, the cell will attempt to repair it. Now let's say our cell has passed the G2 checkpoint and begins mitosis. We have another checkpoint here during M phase, and it occurs before anaphase because once we pull the sister chromatids apart, we can't go back and fix any errors. That's irrevocable. We want to have exactly one sister chromatid from each duplicated chromosome in each of the 2 daughter cells. So the cell will pause at metaphase and attempt to determine whether the kinetochore pair is attached to a microtubule from two opposite poles of the cell. Remember, those kinetochores can sense tension. So cool. And therefore, the cell can actually sense whether the kinetochores are correctly attached. The cell will pause here and won't be able to proceed until the checkpoint is passed. Okay, but maybe by now you're asking, well, what happens if a cell fails a checkpoint? That is a great question. Well, first, the cell will attempt to repair whatever problem it senses. But what if it can't? Well, in that case, the cell will undergo programmed cell death, or apoptosis. By the way, you can pronounce this word apoptosis or apoptosis, depending on how fancy you want to be. The Greek root word tosis has a silent P, but really, either of these pronunciations are commonly used and completely acceptable. Apoptosis is only one of the types of cell death, and it is highly regulated. The cell proceeds through a series of steps that result in it dying and fragmenting into small bodies, which are then engulfed and digested by phagocytes, which are immune cells. Apoptosis is not by any means a rare process. In fact, we estimate that in your body today, there are 50 to 70 billion, with a B, cells that will undergo apoptosis. Apoptosis is also a very normal part of development. What we're looking at here is the development of a mouse paw. You can see that it starts without distinct digits. It kind of reminds me of a flipper, of an aquatic organism. But as embryonic development proceeds, we see digits develop. They do not grow out of the main body of the paw. Instead, the cells between the digits die to release them into separate digits. The same process happens in humans, by the way. Occasionally it fails, which results in fused fingers or fused toes. Apoptosis is also a part of brain development. During embryonic development, we produce too many brain cells, and the extra ones die off via apoptosis. Okay, so to recap, the cell cycle has checkpoints, which are a time when the cell pauses and checks that conditions are right to continue with the cell cycle. Most particularly, detecting and correcting DNA damage so that the cell is not dividing with damaged DNA. If the cell does detect a problem, it will attempt to repair it. But if the damage is irreparable, a normal cell will undergo apoptosis and die so that the damage is not perpetuated. Sometimes this process fails, and cells that are unable to undergo apoptosis are predisposed now to become cancerous.